Howdy, this is Browdy. I'm a little late in making this video, but I wanted to make it all the same, and I'll do my best to make this relatively quick for something that's kind of a long process. And so this is the first of two lectures for the protein synthesis unit, and so this covers proteins, transcription, and translation. So there's a lot to unpack here, so let's jump right in. So first and foremost, you'll see on the notes that there's three types of traits that we really consider in class. And so we have those over here as well. And those are structural, physiological, and behavioral traits. Now structural traits have to do with support and appearance of an organism. So for me, a structural trait could be that I have dark hair normally. I'm short. I have relatively light skin that gets kind of darker complected relatively easily. Anything like that. I have brown eyes. Those would be structural traits. Physiological traits have to do with how an organism functions. So while these don't apply to me, these would be considered physiological traits. So if I were diabetic, that would mean that my body doesn't produce or take in insulin as efficiently as it could. If I were lactose intolerant, that would mean that my body in some way, shape, or form doesn't use or make lactase efficiently or correctly, and so I'm not able to break down lactose. Those would be physiological traits. Also, there's behavioral traits, and so how an organism behaves. So that could be something like being aggressive. If I were to protect others. So behavioral traits we see more along the lines of like other organisms. So we certainly have behavioral traits as humans, but we typically think of behavior as more like an animal characteristic, kind of beyond the realm of humans most of the time. Now, obviously, we could look at humans as well. So I have some examples here as well, but any of those would be considered one of the three kinds of traits. So I'm going to keep on moving. So if those are traits, we have to also think about what makes those traits what they are. And for us, we're going to think about that in terms of proteins. So for example, my skin color is determined by proteins. So melanin, for example, how much melanin I have in my skin, among other pigments, determines what color my skin is. And so that is obviously just one of many factors or one of many examples, but things like that would be proteins being responsible for traits or proteins interacting to create traits. So we can think about the types of proteins as well. Now, one of my favorite acronyms in the whole year, we're going to talk some SHET. So SHET, S-H-E-T, stands for the four basic types of proteins. There are other types that we'll mention, but these are the four that we'll consider the most. These are structural, hormones, enzymes, and transport proteins. So as the name implies, structural proteins have to do with support and appearance. Hormones have to do with communication between cells. Enzymes, which we've talked about before, speed up, or we call that catalyzing, chemical reactions. And then transport proteins move other molecules in some way, shape, or form. And each of these interact in different ways to control different traits. And so some examples of these. Structural proteins, for example, could be keratin, which makes up fingernails or claws. That's what makes them so hard or rigid. Melanin contributes to skin color. Collagen is another example. Hormones deal with communication between cells. So testosterone and estrogen would be examples of hormones. These are the sex hormones, we typically call them, or the male and female hormones, and that triggers all sorts of different responses in your body, kind of based on the presence of one or the other, and those often lead to distinctly male or female characteristics. Adrenaline, which you've probably heard of, maybe like an adrenaline rush, that's a hormone that causes your heart to beat faster, and we kind of relate that to fear and excitement. Insulin is a hormone as well, and so if you're diabetic, what happens is that you either don't produce insulin correctly or your body doesn't use insulin correctly. And insulin is a hormone that signals your cells to take sugar out of the bloodstream, so it controls blood sugar. Enzymes we've talked about before over the course of the previous unit, and we talked about DNA polymerase or helicase. And what you'll notice about enzymes is that they have ASE or ACE at the end of the name. And so enzymes speed up chemical reactions, or we call that catalyzing chemical reactions. So they're a catalyst. And so, for example, DNA and RNA polymerase help copy DNA or RNA. So they help link nucleotides or monomers into a polymer. Lactase is an enzyme that helps break down milk sugars, or lactose. Amylase is in your spit, and that's an enzyme that breaks down carbohydrates or sugars. Some other examples of proteins would be transport proteins. And so we've also talked about these in the cell membrane unit. So when we talked about protein channels that allowed molecules to go from one side of the membrane to the other, that would be a transport protein. But hemoglobin is also a transport protein, and it's a protein in the blood, in red blood cells specifically, that carries oxygen or carbon dioxide waste, and it kind of moves it throughout your body. Immunoglobulin is also a transport protein, and it's part of immune cells with similar functions. So those are our 4 basic types of proteins. Now there are other types of proteins, so Shet plus. We see these down here in the notes as well, and I'll do my best actually to zoom in a little bit here on the notes. That might have helped. I'll zoom in just a little bit more. There we go. I want to make sure that you can see this stuff. I did also send out the notes, so if you're looking for the notes and not sure where to find them, those are all on Canvas. The completed notes are. But so we have other types of proteins. We have storage proteins for stockpiling materials or chemicals or molecules for later use. Receptor proteins for responding to different stimuli or a stimulus. Defensive proteins for protection against disease, and regulatory proteins for controlling which genes are expressed and how much of a protein they make. And so those are just some other kinds of proteins. But we're going to keep going. There we go. So I'm going to keep on, excuse me, got way ahead of myself here. So I'm going to go on to the next page. There we go. Okay. So how does DNA make proteins? There's 2 processes that we're going to talk about, and those are transcription and translation. So first and foremost, what does it mean to transcribe something? Well, it means to make an exact copy. But I like to add a little clause at the end and say an exact copy in a slightly different form. So the example I like to use is right here. Oh, there we go. A cell phone. All of you have some sort of voice assistant on your cell phones if you have one these days, right? I use mine all the time if I'm driving and I need to quick call somebody hands-free or send a text to somebody hands-free. I'll say, hey, so-and-so, call mom or whatever. Or I'll say send a text, and then it'll say, what do you want to say? And I'll say a sentence, and then it will write it down. And that's dictation, but that's transcribing my message from voice to text. In a court case, there's always a transcript of everything that was said. So they take the vocal speech or the verbal speech and make that text. Another example I like to use is back in the old kind of medieval days when there were kings and queens and castles. I'm going to use this analogy a lot. They had a scribe, and if they wanted to send a message to someone else or to something, they weren't going to go, for example, if they were going to go tell somebody, hey, we're going to war with you, they weren't just going to walk over to their castle and knock on the big gate or cross the moat, whatever, and say, excuse me, we're going to war, right? They wouldn't do that. They would say, hey, scribe, come here. And they would say their message to the scribe. The scribe would transcribe it or write it down, and then they would leave the castle and go carry that message. And we're going to come back to that idea. To translate something means to change from one language to another. Oh, excuse me. Nothing like getting an email in the middle of a video lecture. But so, to translate something means to go from one language to another. So here we have just a variety of different ways of saying hello. Now, in DNA, transcription is the process of copying DNA onto a molecule called mRNA, which stands for messenger RNA. So DNA and RNA speak the same language, meaning they're made up of the same things. So nucleotides. Now, you might be going from DNA nucleotides to mRNA nucleotides, but they're still nucleotides that have a sugar, a phosphate, and a base. That's transcription. And so the purpose of transcription is to copy the DNA code onto a piece of mRNA. that mRNA leaves the nucleus, because remember, DNA is in the nucleus, and just like the king or queen would say, hey, scribe, take my message out, right? Because the king or queen's not going to leave the castle or the fortress or wherever they are, or the palace. The scribe is going to do it. The mRNA copies down the code from the DNA so the DNA can stay in the nucleus, and then it leaves to go make proteins. Translation, on the other hand, is the process of changing the DNA code into a new language entirely. So DNA, if that speaks the language of nucleotides or its monomers or nucleotides, proteins speak the language of amino acids, or proteins are a polymer with amino acids as the monomer. And so translation happens at a ribosome, which is in the cytoplasm. So we have this thing that we've mentioned a few times now in multiple units called the central dogma, and essentially the main pathway or main idea of molecular biology or of genetics. And so there's three processes that go along with this. So we had said initially that the central dogma is DNA goes to RNA goes to protein. That's the central dogma of genetics, molecular genetics, whatever you want to call it. And that's true. But we can also associate processes with these things. So DNA, when it copies itself inside the nucleus, is called replication. Getting all kinds of messages. So DNA to RNA, on the other hand, is transcription, which also happens in the nucleus. So DNA to RNA, transcription. RNA to protein, on the other hand, is translation, and that happens outside of the nucleus. Again, I know I'm going relatively quick here. So before we talk too much about transcription or translation, we need to compare DNA and RNA. So DNA stands for deoxyribonucleic acid. Deoxyribose refers to the sugar that it has in its nucleotides. RNA refers to ribonucleic acid because ribose is the sugar in the nucleotides. DNA is typically double-stranded, it's a double helix, whereas RNA is typically a single strand. DNA has 4 bases, adenine, cytosine, guanine, and thymine, whereas RNA has adenine, cytosine, guanine, and uracil instead of thymine. So instead of T's, RNA has U's. And lastly, DNA has only one type of DNA, so there's only one kind, right? RNA has 3 kinds, M, T, and rRNA, messenger, transfer, and ribosomal. So messenger RNA, or mRNA, copies the message from DNA, or the code from DNA, and takes it outside the nucleus, into the cytoplasm. tRNA, or transfer RNA, is a molecule that matches with the mRNA code at the ribosome, and then it brings the correct amino acid. So transfer RNA is a molecule that literally just carries amino acids and looks for the correct set of mRNA to pair with. And we'll talk more about it. Ribosomal RNA, or rRNA, reads the mRNA and holds the tRNA. Now we'll talk about that. So ribosomal RNA is essentially a ribosome. And here's what they look like. So mRNA is the one that we typically think of as RNA. tRNA is kind of a funky molecule that carries an amino acid, and then rRNA is the ribosome. Oh my gosh. I'm getting emails left and right. This is what I get for making a video on Cyber Monday. So judge me all you want for getting the emails that I am. So I'm going to keep going here. Now we have transcription. So I'm going to jump into that. So transcription is a process, again, of copying from DNA into mRNA. So we're just going to go through the steps. So the first thing that happens in transcription, just like in DNA replication, where we had DNA polymerase, we have RNA polymerase. And so it's a protein, which we would know now is an enzyme. It finds the starting point of a gene and it starts to unwind the DNA. And we call that starting point the promoter. So it finds the promoter in the DNA and RNA polymerase starts to unwind. Now you'll see that there's this big yellow blob here, and not only is that RNA polymerase, there are a few other enzymes and proteins as well, and we call that the transcription complex. Now, the transcription complex is using DNA as a template for RNA polymerase to add mRNA nucleotides. So this is happening in the nucleus, and in the nucleus, just like there are free-floating DNA nucleotides, they're free-floating mRNA nucleotides. And so those pair up with the DNA, and the RNA polymerase bonds those free-floating nucleotides together into a polymer. Right, so polymerase means it takes monomers and makes them into a polymer, so that's what we see here. These monomers are getting linked into a polymer. The mRNA strand, once it's formed into a strand, detaches from the DNA and it leaves the nucleus to go to a ribosome, which is in the cytoplasm. So it's the messenger, and then the transcription complex breaks apart. So like I said earlier, like to think of the mRNA kind of like it's like the scribe for a king and queen. So if the king or queen is the DNA, it's the head honcho, right? It's in the nucleus, or they're in the castle or the palace. They're not going to leave that safety. They're going to have a scribe take their message and take it wherever. DNA is not going to leave the safety of the nucleus. It's going to have a messenger take down or transcribe its message and then leave to the cytoplasm to do its bidding, right? Or to make a protein. Now, I'm not sure what happened here with the title of this, but to show you how we would go from DNA to mRNA, if our sequence is TAC, just like we were making a complementary strand of DNA in replication, we would do that with mRNA, but with U's instead of T's. So T would still go to A, like normal, but A would go to U instead of T. C still goes to G, and G still goes to C. So you can use this here to check your answer for this portion, if you would like. Now, I'll talk in a bit about why we read this 3 at a time. And now we're going to move on to translation. So I'm going pretty quick through this, I know, but I want to keep this video as short as I can, and hoping to not get any more emails. So if that's transcription, going from DNA to mRNA, let's talk about translation. So first and foremost, translation creates chains of amino acids, and those amino acids are coded by mRNA base sequences. And so we call 3 letters of mRNA a codon. Oh my gosh. Told you I was going to get another email. And so each combination of 3 letters is a codon, and those code for specific amino acids. And so AUG here would code for one amino acid, and CUU here would code for another amino acid, and those are 3 bases long. So these 3 base codons on mRNA match up with base groups on tRNA, transfer RNA. And on the tRNA, we call those bases the anticodons. So it's not the anticodon that codes for an amino acid, it's just the anticodon that knows which mRNA to drop off the amino acid at. So tRNA is this molecule here that has an amino acid on it, and it brings that amino acid to the ribosome where the mRNA is being read. And ribosomes have 2 parts, one on the bottom to hold the mRNA and one on the top to hold the tRNA. So these amino acids that are carried by the tRNA are what get linked to make proteins. And so there's a few ways you can draw it. We sometimes see it as this kind of like cross-shaped sort of thing. We sometimes see it as this kind of funky, like, I don't know, polygon sort of thing. In either case, they'll be carrying an amino acid in some way, shape, or form, and they'll have an anticodon. So if here's our mRNA sequence and here's the codon that's being read, the tRNA anticodon is how it knows to pair up and drop off its amino acid. But let's look at a little bit more detail. Okay. So there's the ribosome, which is the site of translation, and these are in the cytoplasm, but they have two subunits. They have a large subunit that holds the tRNA, and they have a small subunit that holds the mRNA. Now, the steps of translation. The first thing that happens is it always begins with a start codon, and the start codon is AUG. There's only one of them. And so, just like a computer program might not run until it gets the signal to start running, mRNA won't start getting translated until the start codon is read. And the start codon, again, is AUG, and that codes for an amino acid called methionine, which we'll talk more about. Once that mRNA is in the ribosome, a tRNA molecule with the complementary anticodon comes to join it. So the mRNA is being read here in the bottom portion, and the tRNA comes and drops off its amino acid. And it brings that specific amino acid. So the codon, the mRNA, is what codes for the amino acid. The anticodon on the tRNA is how the tRNA knows to pair up with it. Then the ribosome helps to form what we call polypeptide bonds between the amino acids. So once multiple tRNAs have brought in multiple amino acids, those amino acids get linked together with a polypeptide bond, or just a peptide bond. The used tRNA is then released to go find another amino acid, and the ribosome pulls along the mRNA by another codon. So here we see a tRNA bringing methionine because it's the first amino acid, it's the start codon. Then another tRNA comes in to pair up with this codon. And it comes in, a peptide bond is formed between the first amino acid and the second, and what we see, wrong way, is that that used up tRNA gets ejected, and it happens again and again and again. And once there's a new codon in place, the new tRNA comes into the ribosome, pairs up, drops off its amino acid, and so on. And this process goes on and on and on and on until a stop codon is reached. And it will have made a chain of peptide bonds or amino acids, and we call that a polypeptide, which is essentially a protein. But it will do this until a stop codon is reached. Now there's a few of those, but just like a computer program will stop running when it reaches the stop command, once stop is reached by the ribosome, it just stops doing what it's doing and it lets go of the protein. Now we'll talk about the stop codons in a second. So to clarify something from earlier, we said that the central dogma is DNA goes to RNA goes to protein. But now we know that, a little more specifically, DNA gets transcribed into mRNA codons. Then with the help of tRNA anticodons, that is translated into protein in the form of amino acids. Now we've said that AUG is the mRNA start codon, but there are also three mRNA stop codons, UAA, UAG, and UGA. Now these don't code for an amino acid, so AUG, the start codon, does. It codes for methionine. These don't code for an amino acid, they just signal to stop. So if we were to go through this whole process of going from DNA to mRNA, first we would take the DNA sequence and transcribe it to mRNA. Now hopefully that's something that you can do at this point. So T goes to A goes to U, C goes to G, G goes to C, right? Now the mRNA is going to pair up with the complementary tRNA. So these are the anticodons here. And you might notice that because this is complementary to this and this was made complementary to this, that the DNA and the tRNA are the same except the T's are U's. It's a nice thing to notice. But the mRNA are how we figure out the amino acids, but how do we actually do that? We use something called a gene wheel or a codon wheel. And so we can look up the code. And so in our example that we just looked at, AUG was the start codon. So what we do, the way we read this, is we start with the first letter of our codon in the center here. We read this from inside out. So we first find A. Okay, and we've narrowed it down. So then we can ignore the rest of the wheel. So we're just looking at this section over here. A, and then we go to U. So we go down here to U. So we can ignore all these. We go A, U, G. And that shows us methionine. So here we have methionine. And then if we keep going through our sequence, we do the same thing over and over. GCG gives us alanine. So we say, okay, GCG gives us alanine. AAU gives us asparagine. So AAU. And so on. And we do this until we get to UGA in our sequence. UGA. And that says stop. Now what you might notice is that there's what we call redundancy in this code. So for example, UCU, UCC, UCA, and UCG all give us serine. So all four of those different codons, all four of those different codons give us the same amino acid. And that's just something to be aware of because there are 20 different amino acids, but 64 possible combinations of these four letters if we're looking at them as three bases. So there are three different stop codons, UGA, UAA, and UAG. Those are all stop, but only one start, AUG, and that's methionine. And so we would then fill in here methionine, alanine, and so on until we got to stop. Now these three-letter abbreviations, you can find those on the codon wheel downloadable page that I put up on Canvas. So if you haven't seen that yet or gotten a physical copy, please check that out and you can use that for this. And I believe that's all for this video lecture. So I apologize for the text message and the emails. So maybe that gives you a slight look into the sorts of emails and texts that I get. But hopefully this is helpful and has been helpful for you all. If you have any questions, as always, just shoot me a Canvas message or ask me in class, send me an email, whatever it is that can help. I hope this is helpful for you. Sorry that we're almost at half an hour. But best of luck with all of this, and I will see you next time. Take care.